Let's quickly review gene expression. First of all, our RNA polymerase II in the nucleus copies the template strand of the DNA to produce a primary RNA transcript. The primary transcript in eukaryotes is processed by addition of a 5' GTP cap and a poly A tail on the 3' end. The mature messenger RNA is then exported through the nuclear pores into the cytoplasm. Here, the 5' cap of the messenger RNA associates with the small subunit of the ribosome, and the initiator tRNA sets itself in place. The large subunit is added, and now we have an initiation complex. The ribosome cycle begins with the growing polypeptide chain attached to the tRNA in the P site. The next charged tRNA binds in the A site, or the arrival site, with its anticodon, complementary to the codon on the mRNA in this site. A peptide bond forms between the amino terminus of the next amino acid and the carboxyl terminus of the growing peptide chain. So the chain is transferred to the tRNA in the A site, leaving the tRNA in the P site empty. The ribosome then translocates. It moves over one codon towards the 3' end of the mRNA. This moves the growing chain into the P site, leaving an empty tRNA in the E site and the A site ready to bind the next charged tRNA. This cycle continues until we have the full amino acid chain. This amino acid chain will end once we reach the stop codon. A termination factor lands in the A site, and the whole thing blows up. Let's take a moment now to look at mutations. Mutations are altered genes. Point mutations alter a single base. We can have a number of different point mutations. In base substitutions, we substitute one base for another base. You can see that each of these different substitutions has a different effect. In a silent mutation, the same amino acid is inserted. In this case, we end up swapping a C for a G. However, when we look at the genetic code, this still codes for proline, so there's no change from the original sequence of amino acids. In a missense mutation, we see changes in the amino acid that's inserted. Usually this will involve the first or second position of a codon. So here, if we change the A to the T, then we'll see that histidine is put in this place instead of threonine. The next kind are nonsense mutations. This is when we change the codon from a coding codon to a stop codon. That means that there is no further addition of amino acids to the polypeptide chain. Here's an example with the synthesis of hemoglobin and explains in detail what's going on with sickle cell hemoglobin. Simply, there's a base substitution. In the normal hemoglobin sequence, there's an adenine right here. In the abnormal sequence, there's a thymine. This causes a difference in the amino acid that ends up at this codon. It should have been glutamine. However, it ends up being valine. This is a big switch because we switched a polar amino acid for a nonpolar or hydrophobic amino acid. That results in abnormal tetramer for hemoglobin. So these two beta subunits are unable to carry oxygen. And as they stack together much more tightly. And this distorts the normal red blood cell shape into a sickled shape. Let's look at a few different mutations. A Frameshift mutation results from the addition or deletion of a single base. It has much more profound consequences because it alters the reading frame. Here you can see the loss of one nucleotide. It causes all the amino acids downstream of that nucleotide to be altered, and thus certainly alters the function of the protein. Here we could have an insertion. The insertion of U at this location causes the coding of a stop codon. So this would be a frameshift that causes nonsense. If you have the insertion or deletion of 3 whole nucleotides, then there's not an extensive frameshift. Just one amino acid will be changed in the whole sequence. One of the first genes isolated was associated with the human genetic disease called Huntington's disease. The gene for Huntington's disease provided a new kind of mutation. This gene contains triplet sequences of DNA that is repeated. And this repeat unit is expanded in the disease allele relative to the normal allele. Since this initial discovery, at least 20 other human genetic diseases appear to be due to this mechanism of triplet repeat sequences. The last type of mutations we'll consider are chromosomal mutations. This is when we have a radical change in the structure of a whole chromosome. There can be deletions, where part of a chromosome is lost entirely, or duplications, where part of a chromosome is copied. There can be inversions, where part of it is inverted and put in reverse order. And then translocations, where part of the chromosome is moved to an entirely new location. Obviously, these mutations can have some profound effects. Mutations are the starting point for evolution. Too much change, however, can be harmful. An individual with a greatly altered genome may not be able to survive right here and now. So balance has got to exist between the amount of new variation and the health of a species. So this concludes our chapter on transcription and translation, or gene expression. Let's take a quick card quiz to finish it off. Substituting an adenine nucleotide for a guanine nucleotide would cause a frameshift mutation. Is this true or false? A codon AUG matches with which anticodon? A substitution mutation changes the codon CCC to CCA. Both of these codons code for proline. What type of mutation is this? Here are your answers.